Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 9th February 2021. As we are into the revision mode, we are revising the entire syllabus in the form of CBSE sample papers, and we strictly follow the new guidelines that have been given by the CBSE for your batch. And yesterday's session we answered section A in this sample paper. Now we are going to section B. Now I am going to give you questions and answer. Now section B, consists of 9 questions and each has got 2 marks. So 9 2 ja 18. Section A 22 marks. Section B 18 marks. So 22 plus 18, 40 marks out of 70. Keeping that in your mind, I have advised you also keep separate notebook for this. All the 11, 12 sample papers I am giving, you must write separate notebook so that it will be very handy during your board exam. Section B, question number 17. Sunita's Babi is not allowed to enter the kitchen during the days of her menstrual cycle, MC. Sunita's mother thinks that she is impure and dirty, and the food prepared by her is also unhygienic. Give your opinion about such a traditional belief. Now the answer. Menstruation is a biological and natural phenomena that occurs to each woman. Menstruation is the process of shedding of the uterine lining on a regular monthly basis. It begins at puberty and is the reproductive cycle of the female body. Every month, the uterus prepares itself to receive a fertilized egg. So in anticipation, this happens. However, due to lack of education in many parts of the country, people practice such a cult practices, for example, not letting them enter into the kitchen. Awareness plays an important role here. Small groups in the society should be set up to make people more aware about the phenomena. What is important is whether there is menstruation or not, cleanliness. So if the person, like Sunita's Babi, if she is maintaining cleanliness and biologically clean, then there should not be any problem because it is a natural phenomena. Question 18. A child has blood group O. If the father has blood group A and mother blood group B, work out the genotypes of the parents and the possible genotypes of other offsprings. Here it is genotype. Since the blood group of the child is O, so it must be homozygous for the allele IO or I0. Since the parents have blood groups A and B and produce a child with O blood group, they must have to contribute the I0 gene and be heterozygous. That is, the genotype of the father must be IA I0 and the genotype of the mother is IB I0. So the genotypes combination, the permutation and combination is IB I0. In the case of father, IA, it should be IA IB, one combination, another combination IA I0. So the possibility of getting O group is in 2 ways. One is the IB I0, another is I0. Then this possibility will come. Whether like I0, both of them, straight away it is 0. That is O group. IO, straight away O group. Like father is IO, and mother is IO. No doubt the child has to be O group. But here the father's group is A, so the choice is IA. And mother is B group, so IB. So the combination is this. This is the genetic combination where the child is becoming 0. That means heterozygous. This is how you have to work out the combination. Question 19. Write short notes on the production of human growth hormone by E. coli. Answer. Human growth hormone HGH is very useful to children born with hypopituitarism, which is a form of dwarfism caused by undersecretion of HGH by the anterior pituitary gland. It also helps in the healing of injuries. The gene for HGH is introduced in the plasmid of bacterium E. coli. This recombinant E. coli bacterium secretes human growth hormone, which is used for treating hypopituitarism. Question 20. With respect to understanding diseases, discuss the importance of transgenic animal models. Transgenic animals can serve as models to understand progression of various diseases in human beings. Transgenic models exist for many human diseases like cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. These animals can be observed to understand the causative gene for a particular disease. This can help in devising suitable treatment for a particular diseases. Normally we use for any experiment, science experiments, we use animals. Here we are using transgenic animals. The choice question is, how has recombinant technology helped in large scale production of vaccines? Explain giving one example. Recombinant vector vaccines make use of recombinant DNA technology or DNA technology. These vaccines make use of an attenuated virus or bacterium to introduce microbial DNA to cells of the body. That's why we are using it rDNA. Recombinant technology has allowed the production of antigenic polypeptides of the pathogen in other microbes like yeast and bacteria. For example, hepatitis B vaccine is produced using yeast cells. Question 21. Based on your understanding of genetic code, explain the formation of any abnormal hemoglobin molecule. What are the known consequences of such a change? Normally, human beings have following types of hemoglobin: HbA, HbA2, HbF. Alteration in genes for beta chain on hemoglobin results in the formation of HbS type of hemoglobin. This type of hemoglobin molecule is responsible for sickle cell anemia. In fact, S stands for sickle cell anemia. Question 22. How does one visualize DNA on a agarose gel? DNA fragments separate when they are moved towards the anode in the electric field. Agarose gel provides the matrix through which DNA fragments separate due to sieving effect. Separated DNA fragments can be visualized only after staining with ethidium bromide and then by exposure to UV radiation. After staining, DNA fragments appear as bright orange bands. And now there is a choice question. A recombinant vector with a gene of interest inserted within the gene of alpha galactosidase enzyme is introduced into a bacterium. Explain the method that would help in selection of recombinant colonies from non-recombinant ones. The method is based on color reaction, that is blue-white selection. The alpha galactosidase enzyme can cleave, means break, colorless synthetic substance X-gal into a blue colored product if the gene is inactivated by insertion of gene of interest into it. The development of blue color will be prevented. Question 23. Explain, taking one example, the effect of co-extinction in biodiversity. Co-extinction means that when a species is going to become extinct, the plant and animal species associated with it in an obligatory relationship also gets, that's why co-extinction. For example, Co-evolved plant pollinator shows mutualism, as in the case of Pronuba ecacilla and yucca plant. If one partner is on the verge of extinction, the other partner will also get extinct because it is mutualism. Question 24. How is Cuscuta adapted to be a parasitic plant? Cuscuta has lost its chlorophyll during evolution and developed haustoria, through which it derives nutrition from host plant. Thus it is adapted to parasitic mode of life. Question 2. Why do cattle avoid grazing on calotropis plants? Explain. Calotropis plants produces poisonous cardiac glycosides. Therefore, cattle or goat do not graze on these plants. Question 25. The major cause of species loss. Question is, what are the major causes of species losses in a geographical region? The major causes of species losses in a geographical region are: 1. Habitat loss and fragmentation. 2. Overexploitation of natural resources. 3. Alien species invasions and or introduction of exotic species. And of course, just now we discussed co-extinction of related species. So this is how you need to answer to the point. And in our next session, we will answer section C and D. Until then, keep practicing. As I again tell you, keep a separate notebook for this. And the good news is that you are coming back to the school. It will be very, very helpful once you sit for self-study. Now there is no time for us to discuss at length. We need to be specific. That is the reason why I am telling you, keep this separate notebook. Until then, keep practicing,